Hi everyone, welcome to Microbial Genetics Lecture 3, where we're going to keep talking about chromosome structure, replication, and segregation. So before we start, there's a few quick announcements. Don't forget that after this lecture, you need to go to the discussion board on Canvas and write one thing that you learned and one question that you still have about this lecture. Quiz 1 should also be live after class. Don't forget you have 24 hours to take this quiz, so you need to take it before 10 a.m. tomorrow morning. Okay, let's move on to chromosome organization and cell division. So the last two lectures, we talked about replication of the bacterial circular chromosome. So the question is, what happens after DNA replication? Well, before the chromosomes can be partitioned into daughter cells, they first need to be separated from each other. So there's a couple problems that can occur during cell division that will inhibit chromosome segregation. So for example, chromosome dimers are actually fairly common. This is where sister chromosomes are covalently linked, forming one large DNA molecule. So you can see here, what should be two chromosomes is one big chromosome. This often occurs through a recombination event, shown here by this red X, that occurred during replication, or the chromosomes can become otherwise entangled. There are two solutions to this problem: general recombination to form two DNAs, or site-specific recombination involving the XER-CD system. It's this XER-CD and diff site system that we'll talk about today. So this system is found in E. coli, and there are very many similar mechanisms found in many other bacteria. First, let's define recombination. We'll have a whole lecture dedicated to this later in the semester. But for now, it's the process by which genetic material is broken and joined to other genetic material. That genetic material can be multiple different chromosomes or different regions of the same chromosome. First, let's take a little concept quiz. XERC and XERD are proteins, and the diff site is a site in the E. coli chromosome. Are XERC and D and diff site cis or trans acting factors? XERC and XERD are diffusible proteins and are therefore trans-acting factors. Remember, they can work on any molecule of DNA. However, the diff site is a DNA sequence and therefore can only act on the piece of DNA which it is on, making it a cis-acting factor. So if we do end up with a chromosome dimer, so you can see in this picture here, there's two chromosomes. One is the dark line, one is the gray line, meaning that you have two diff sites. If two diff sites are present on one piece of DNA, XERC and D will promote recombination specifically at those diff sites to resolve that chromosome dimer into two sister chromosomes. If we look at the E. coli genome map, we can see our OREC positioned here at 0 degrees, meaning that TER should be about 180 degrees away from it. And you can see that the diff site is located here near TER. Why do you think that is important? It's important because the sequences near TER are the last to be replicated. Therefore, the diff site could only be present in two copies if the chromosomes have dimerized. So if we look at this image here, if the diff site were near OREC, you would have two diff sites on one piece of DNA very soon after DNA replication has begun, but before the chromosome has been completed, meaning that the XERCD system might try to resolve this chromosome before replication even finished, which could be bad. So the XER-CD recombination system is coordinated with cell division through a protein called FTSK. FTSK is a DNA translocase. That means it can pump DNA to move or align the diff sites to mid-cell. And this facilitates DNA segregation. So if we take a look at these three images, the septum starts to divide. So that division septum here, you can see that cell's pinching mid-cell. And right there at that septum, there is this protein called FTSK. So you can see here it's labeled. In this particular example here on the left, FTSK trapped only one of the sister chromosomes. So it needs to move the chromosomes over to the right to align them where the diff sites are positioned right in the middle of the cell. So you can see in these different examples, FTSK needs to pump or move the DNA in different directions in order to align the diff sites in one position. Once it does that, it can recruit the MICR3300 proteins, which will then resolve those chromosome dimers. Those sister chromosomes are now free from each other, and the cell can divide in the middle into two identical cells. So how does FTSK work? How does it know directionality? Well, there are sites known as KOPS sites. These stands for FTSK, orienting polar sequences. They're oriented in such a way that the DNA can only be read in one direction. So it will pump the DNA towards the diff site close to the terminus. Okay, so that was the XERCD system. So that is to facilitate chromosome dimers. Another potential problem that can occur during DNA replication are interlinked chromosomes. And we're going to talk about topoisomerases, how they function in general, and also how topoisomerases can function in this process called decatenation to resolve torsional stress ahead of the DNA replication fork. Okay, but before we talk about that, we need to talk about supercoiling or the supercoiling status of the DNA. So if we look at this figure A here of a relaxed piece of DNA, we see the double helix that has a typical right-hand twist. If you hold either end of that DNA molecule and twist it in one direction or the other, you can induce either positive or negative supercoils. Positive supercoils are defined as an overwinding of the DNA, and negative supercoils are defined as an underwinding of the DNA. Within the cell, the DNA is negatively supercoiled, meaning that it's underwound or twisted in the opposite direction to the Watson-Crick helix. On average, there's a negative supercoil present every 300 bases in the E. coli genome. This supercoiling causes the DNA to wrap upon itself, resulting in compaction of the chromosome or DNA condensation. So if we take a look at some of these other images here, in B you have a linear piece of DNA, and if you grab one end and twist it, it's going to be free to twist on the other end and you won't get any supercoils. However, if you constrain both ends and then twist, now you can see the formation of supercoils. Similarly, if you have a circular chromosome with a nick in it, you won't be able to induce any supercoils. However, if you have a closed circular DNA molecule, you can twist and induce either positive or negative supercoils. This is important because DNA replication generates supercoils. So remember our DNA helicase encoded by the DNA B gene product opens up that double helix so that each strand can serve as a template during DNA replication. And the pulling apart of those two strands of the helix induces these positive supercoils ahead of the replication fork. This increases torsional stress, and if not resolved, will cause the DNA polymerase to get stuck and won't be able to complete that round of replication. Fortunately, the cell has mechanisms to resolve this torsional stress through an enzyme called DNA gyrase that moves ahead of the fork and removes positive supercoils. And before I tell you more specifically about how DNA gyrase functions, we need to know that it's a class of enzyme called a topoisomerase. Topoisomerases facilitate DNA supercoiling, meaning they can introduce or remove DNA supercoils. So they can remove supercoils by breaking one or both strands of the DNA, passing the DNA through the break and resealing that break. There are two types of topoisomerases. Type 1, which only break one strand of the DNA, or type 2, which breaks both strands of the DNA. So here we're starting with a molecule of DNA that has two supercoils. If we follow this top image here, this is an example of a type 1 topoisomerase. It's going to break one strand. It's going to pass one strand through the other and reseal it. So type 1 topoisomerases can remove one supercoil at a time. If we look at the bottom example, this is an example of a type 2 topoisomerase, where you can see that both of the DNA strands were cut, they're passed through each other and then resealed. Type 2 topoisomerases can either add or remove supercoils two at a time. So you can see we started with two supercoils and we ended with none here. Okay, so DNA gyrase is a type 2 topoisomerase. It's made up of two subunits, gyre A, which breaks the DNA, and gyre B, which has ATPase activity, meaning it can bind and hydrolyze ATP to provide energy for this topoisomerase activity. So if you remember, as the helicase is pulling apart the DNA strands, it's inducing positive supercoils ahead of the replication fork. So gyrase moves ahead of the replication fork and induces negative supercoils to counteract the positive ones made by helicase. So you can see in this particular example, gyre B is going to grab one section of the double-stranded DNA. Gyre A is then going to induce that break by cleaving both strands of the DNA molecule. It's going to hold the two ends apart while passing, you can see here, this dark purple strand from on top to below. Remember, it's using the power of ATP hydrolysis to power all of this. It's going to reseal them once it's passed that strand through it, removing these supercoils two at a time. Okay, so now that we know how DNA gyrase works and how topoisomerases work in general, we can talk a little bit more about a second mechanism to facilitate chromosome segregation, which is decatenation. So the formation of catenates are DNA that become joined together or interlinked like links on a chain during DNA replication. So this is also formed by the unwinding of the template DNA strands by DNA helicase. So you can see here there's so much torsional stress that it passes through the replication fork and causes these two strands, or what should be two strands of DNA, to become interlinked with each other. So basically DNA gyrase either isn't moving fast enough or can't work efficiently enough to counteract the force of DNA replication. Once these catenates are formed, the only way to resolve them is by breaking both strands of DNA, so meaning it requires a type 2 topoisomerase. This function is performed by topoisomerase 4. In this last bullet point here, topoisomerase 4 interacts with FTSK to coordinate cell division and chromosome segregation, very similarly to how FTSK is functioning with the XERCD system to facilitate resolution of those chromosome dimers. Okay, so here you can see in this figure, here's the replication fork, also known as the replisome. And here you have the formation of catenates. So the black line is one sister chromosome, the blue is another, and you can see here they're interlinked. So the cell starts to begin to divide. FTSK is that DNA translocase that's associated with the septum. It's interacting with topoisomerase 4, which can break both strands of DNA and to begin to resolve these catenates before the cell completely divides, meaning that each daughter cell will get one copy of the chromosome. Okay, so let's look at our very simplified bacterial cell cycle. Step one is we need to replicate the chromosome. We need to go from one molecule of DNA to two molecules of DNA. If any problems occur, those two chromosomes need to be separated from each other. So we talked about the XERCD system, we talked about gyrase, and we talked about topoisomerase 4 as three different mechanisms that could potentially help resolve any issues that occur. Then the DNA needs to be segregated to the daughter cells before the cell can divide right in the middle, creating two identical daughter cells. Okay, so let's talk a little bit about DNA organization within the cell. So we're going to talk a little bit about the bacterial nucleoid. The nucleoid is a term that we use just to call the bacterial chromosome. And so bacteria actually coordinate the position of the chromosome within the cell. So if we look, or if we think about the dimensions, E. coli is about 2 microns in length, or 2 micrometers long. However, if we think about the E. coli genome, or the DNA housed within this 2 micron length cell, the genome, if you stretch it out, takes up about 2,780 microns. So it's much longer than the cell is itself. So how does the cell take all of that DNA and all of that genetic material and fit it into this teeny tiny cell? And if you can see in this image here, this is an image of a lysed E. coli cell, and all of these squiggly lines are all the DNA that was inside the cell now being extruded outside of the cell. Well, the cell coordinates the position of the chromosome by holding it in a series of 300 to 500 loops of DNA to form a condensed region. So chromosome condensation is a way to manage chromosomes. It's also a way to condense them after DNA replication and is often coordinated with decatenation. So if they're condensed, the cell or the DNA won't overlap nearly as much and they're less likely to become interlinked. This is similar to chromosome condensation that occurs before mitosis in eukaryotic cells. So some of the proteins that perform this function are called condensins, or proteins that help condense the DNA. These are also referred to as SMC proteins, standing for structural maintenance of chromosomes. They're dumbbell-shaped proteins. They have a globular end with a long coiled region holding them together. So if we look here, the protein from N to C terminus, it has globular ends at the N and C terminus, and then they have this coiled region in the middle. It'll fold up on each other, so the N and the C terminus come together to form this head region. And then there's this hinge region, again connected by that coiled domain. This is one SMC protein, this dark pink, and then in light pink is a second SMC protein. They'll come together to form an SMC dimer, where you can see here they're interacting here at the hinge region. These head regions are where they combine to DNA. And you can see in this other image here, you have these hinges here, you have the heads are binding to DNA, and they can start holding the DNA, looping them and condensing them together in a variety of different structures. So chromosome condensation also helps facilitate supercoiling stress. So wrapping DNA around a protein can relieve supercoiling stress. And remember, this is important because positive supercoils can lead to the formation of catenates, and these can only be removed by topo 4. And so you can see here, as Topo 4 is decatenating these two sister chromosomes. It's also coordinating their condensation with this SMC protein, also known as MUC-B. Okay, so now we've learned how to replicate the bacterial chromosome, how to keep them from being either dimerized or interlinked. So now we're ready to continue with our cell cycle, which is to make sure each of those sister chromosomes are in daughter cells. So how are chromosomes segregated to daughter cells during cell division? Not a lot is actually known in bacteria. This is a big gap in our knowledge about the bacterial cell cycle. In eukaryotic cells, there's a spindle apparatus. However, there's no spindle apparatus identified in bacteria. However, I will tell you about what we do know about chromosome segregation. And part of that includes DNA replication, and then I'll also tell you a little bit about the PAR-AB system. Okay, so I mentioned that our DNA is actually very highly organized within the cell. So if we look at this cartoon here, our origin of replication is this green dot. The origin of replication, or ORAC, always begins in the middle of the cell. So the replosome are these two kind of bluish structures here in the middle. This includes our two molecules of DNA polymerase that will replicate each the leading and lagging strand. And remember, chromosome replication is bidirectional around the circular chromosome. And what happens is, as the origin gets replicated, it starts to move out to the bacterial cell poles. So the ends of the cells are referred to as the poles of the cells, meaning that the terminus, or the sequence that gets replicated last, stays near the middle of the cell. So this image here that I'm showing you, this cartoon image, is based on real data where the origin was labeled with a green fluorescent protein and the terminus was labeled with a red fluorescent protein. So you can see the cell outlined here in black, and the two origins are moving out towards the ends of the cell, and the terminus remains near the middle. So this means that once the cell is done replicating, they're already in a pretty good position for cell division. However, for some bacteria, this isn't sufficient, and they require an active mechanism to segregate the chromosomes to daughter cells. For this, we have the PAR system. It's required for the segregation of chromosomes as well as many plasmids. And so while some bacteria have the PAR system, there are many that don't, meaning that we still don't know a lot about how chromosomes are segregated during cell division. But the PAR system requires three components: PAR A and PAR B, which are proteins, meaning that they are trans-acting factors, and PAR S, which is a DNA sequence, meaning it's a cis-acting factor. So here, PAR S is actually labeled the centromere here in blue. So PAR B, this green protein, is going to bind to PAR S in the chromosome. Here you can see after replication, there's now two of them. PAR A has ATPase activity, meaning it's able to bind to ATP, hydrolyze ATP, and this causes it to polymerize and depolymerize. And as PAR A polymerizes and makes this filament, it pushes the two chromosomes to opposite ends of the cell. Once it's done that, it can depolymerize, allowing the cell to divide. Okay, so now we have replicated the chromosome, we've resolved any chromosome problems, we've segregated the two chromosomes to daughter cells. Now we're ready for the cell to physically divide into two. One of the most critical proteins for cell division is known as FTSZ. It's the bacterial tubulin homolog that forms the Z ring at mid-cell. This is the site of cell constriction and division. And basically what FTSE does is it recruits the proteins necessary for cell division and septation. So if you think about everything that needs to happen, here at the mid-cells, the cell needs to physically pinch and divide, meaning that new cell wall and cell membrane material need to be synthesized right here at that septum in order to separate the two daughter cells. This is actually an image that I took on a super resolution microscope that was able to give me 3D resolution. So in red is our Synechococcus elongatus, the one we're using in the lab. And in green, I have FTSZ that I've labeled. And you can see that really beautiful ring forming right in the middle, and that's where the cell is going to pinch and divide. And here's just another quick example, again, related to what we're doing in the lab section. But if I have that FTSE fused to a yellow fluorescent protein, so here it's kind of looks a little bit yellow, you can see our wild type cells are nice short rods, and you can see them dividing here in the middle. And our ClipX mutants, remember we learned last week in the lab section that they have a cell division phenotype, meaning that they have long cells and they also have an inability to form these FTSZ rings. So FTSZ can't form, the cell can't divide, they get really long and filamentous. Okay, so how does FTSZ work? It's a GTP binding protein. So in the GTP bound state, it can form a filament. So here you can see FTSE bound to GDP, or the diphosphate form. It kicks that out, binds to GTP, changes conformation here from the circle to the diamond, and now it can form a filament. That filament is important for forming that ring around mid-cell and also important in the constriction. So you can see here in this picture, once it's gone from one copy of the chromosome to two, that Z ring or FTSE can begin to polymerize as a ring in the middle of the cell. Okay, so first FTSE needs to nucleate near mid-cell, tether. It needs to stabilize and form a mature ring. Once it does that, it can begin to depolymerize and cause constriction of the cell, ultimately dividing that one mother cell into two identical daughter cells. And filament assembly and disassembly is mediated by whether or not FTSE is bound to GTP or GDP. So again, in the GTP bound state, this is the maroon circles, it can begin to polymerize. So this is important for this nucleation, the stabilization stages here. Once that ring is formed, it can start to hydrolyze the GTP, so hydrolyzing that lost phosphate bond to GDP. And this causes depolymerization, which is then going to cause that constriction of that ring. Okay, so how does the cell know where to form the Z ring? How does it know where the middle of the cell is? I'm going to tell you 3 mechanisms that help to tell the cell where the middle of the cell is and where the Z ring should form and where cell division should occur. So we're going to go over the nucleoid occlusion mechanism, the MIN system, as well as the protein called MIPZ. So first I'm going to tell you about nucleoid occlusion. Remember, nucleoid is another word that we use for the chromosome or for the DNA. So nucleoid occlusion means that FTSE cannot form a ring over the DNA. So what happens is our DNA is here in blue, and there's a protein called NOC. And it binds to our DNA and surrounds that DNA or the chromosome, and it actively inhibits FTSE. So it's going to inhibit it from binding to GTP, and it won't be able to form a ring wherever there is DNA. This is a mechanism that works in our 2 favorite model systems, E. coli and B. subtilis. In addition to the nucleoid occlusion system, many bacteria additionally have the MIN system. It's the MIN-CD complex that inhibits FTSE assembly. It works by having the min C and D proteins oscillate from one pole or one end of the cell to the other pole or the other end of the cell. This creates the lowest time average concentration in the middle of the cell. So if min C and D are inhibiting FTZ from forming rings at the poles, that leaves only the mid cell now as a place where FTZ can assemble and divide. So if you look at this video here, this is one that I took in SE Longatus. Remember, the red is the autofluorescence from the photosynthetic pigments in the membrane. And in green or yellow, I have made a fusion of min C to YFP. And so what you're seeing here is this is taken every 5 seconds. You can see that min C is going from one end of the cell to the other very rapidly. And you can see in that inflection point right there in the middle is where the cell is going to ultimately divide into two daughter cells. Although this video wasn't taken long enough to see many cell division events. Okay, so let's take a quick concept quiz and test what we've learned so far. What would happen to a bacterial cell that was missing the min system? Additionally, let's think about what would happen to a bacterial cell that was missing the NOx system. For a cell that was missing the min system, remember, min allows FTZ to be inhibited at the poles of the cells. So what would happen is you would see polar divisions, so both at the mid cell and at the cell poles, and you would get what we call anucleate cells, or these teeny tiny little cells without any DNA inside of them. These would not be viable. Alternatively, if the NOx system were mutated, but again the min system is still functional, you would not be able to see divisions at the poles because the min system is functional. However, you would now be able to see divisions at mid-cell. In addition, you would see divisions over the DNA. This would end up cutting or shearing the DNA, and if, let's say, this division were to occur over this left chromosome, that cell would ultimately die. And so in most bacteria, the min and the NOx system work together to inhibit divisions at the pole and over the DNA, limiting cell division to only this DNA-free region at the mid-cell. However, there are some bacteria that don't appear to have either min or NOx systems. Alternatively, they have a protein called MIPC. This is an example of another major model organism called Caulobacter crescentus. Called crescentus because of this very beautiful crescent shape. So like I mentioned, they have MIPC. MIPC functions similar to the min system in that it creates a gradient from one end of the cell. However, it doesn't oscillate. So MIPC is located at one end of the cell, or actually at both ends of the cell, and you can see how it's highest at the ends and lowest in the middle. And so if it's lowest in the middle, that is again where FTZ will be allowed to form that Z ring and divide. So with that, our objectives for today are understanding the mechanisms by which chromosomes are separated after DNA replication. So we talked about the XER-CD recombination system, or site-specific recombination system, to resolve chromosome dimers. We also talked about DNA supercoiling by topoisomerases, and how DNA gyrase and topo4, both of which are type 2 topoisomerases, can help resolve interlinked chromosomes. We also talked about how both of these mechanisms are coordinated with cell division, primarily through the FTSK DNA translocase. After this lecture, you should also understand the mechanisms by which chromosomes are segregated after DNA replication. So we talked a little bit about how the DNA is positioned within the cell and how it's organized during cell division, with the origins moving out towards the poles of the cells and the terminus staying in the middle. And we also talked about the PAR-AB system, which is an active
segregation mechanism that pushes the two chromosomes away from each other and towards the poles of the cells. In the last step of the bacterial cell cycle, or cell division, we talked about how cell division, or how the septum forms by the formation of the FTZ ring at mid-cell, and how the MIN system, the NOx system, and the MIPZ systems all function in different bacteria to position FTZ to ensure that FTZ forms in the middle of the cell and that it doesn't form over the DNA. And so with that, please don't forget to write on the discussion board as well as take quiz 1, and I will see you all later in lab.